Okay, welcome everyone to my first tutorial video in cell biology. In this video I will be talking about the cell membrane. So I'll be defining the cell membrane, and I'll be talking about the structure and function of cell membranes lipids. So I'll be focusing on only lipids in this video. There are also other structures like proteins and carbs, but I will discuss them in future videos. So first, what is a cell membrane? What is the general definition of a cell membrane? So a cell membrane is a biological membrane which separates the internal environment of the cell from the external environment of the cell. So if we look here, the internal environment of the cell is represented by the cytoplasm and the other different components of the cell, like the different proteins, ions, organelles, like the mitochondria, endoplasmic reticulum, nucleus, etc. And the external environment of the cell is represented by the extracellular matrix. So we have other cell types, for example. We have connective tissue and the different components of the connective tissue, like proteins, carbs, lipids, etc. And that's the cell membrane. So it separates these two environments. And this separation helps the cell functioning normally, because if we don't have a cell membrane, or if the cell membrane is damaged, then the components of the cell will be leaking out, and therefore the cell won't be able to function normally, or there won't be a cell in the first place. So it's mainly composed of phospholipid bilayer and proteins. So we have here, you can see the proteins which are attached or inserted differently within the cell membrane. We have also, as I said, phospholipid bilayer, because if you look here, then we have two different leaflets. One is facing the outer side of the cell, and one is facing the inner side of the cell. Hence the name bilayer. And the majority of these structures here are called phospholipids, hence the name phospholipid bilayer. But there are other also components, like carbs, for example, you can see here, mainly attached to proteins and facing the outer side of the cell. We have also lipids, other types of lipids, like cholesterol, for example, or glycolipids. So not only phospholipids. So as I said, we will be focusing in this video on lipids and the different structures and functions. Excuse me. So there are three major components of the cell membrane, the three major lipid components of the cell membrane. So if we extract all the lipids that build or are found in the cell membrane, then all of these lipids will mainly be one of these three different classifications. So they could be phospholipids, they could be glycolipids, or they could be sterols. The majority of lipids are phospholipids. About 60% of these lipids are phospholipids. The other 40% belong to either glycolipids or sterols. Now, what is the difference between all of these three? We will discuss it within the next few slides. So phospholipids, we can classify them further into two main groups. Okay, so they could be either glycerophospholipids, which means that they are phospholipids attached to something called glycerol, or they could be sphingophospholipids, which means that these are phospholipids attached to something called sphingosine. Okay, now the four main examples or types of glycerophospholipids are, you might have heard of some of them, Phosphatidylnositol, we have Phosphatidylcholine, Phosphatidylserine, or Phosphatidylethanolamine. So these are the three or four major types or examples of glycerophospholipids. phospholipids. There are also other three, but these three are usually variations, either variations or really rare and exist in other cell types other than animal cell types. For example, phosphatidylglycerol is mainly found only in the lungs, specifically in the alveoli. We have phosphatidic acid, which is mainly found in photosynthetic cells. And we have diacylglycerol, or DAG, which is a variation of phosphatidylinositol. So it's a product of the metabolism of phosphatidylinositol. But in this video, I will only discuss and focus on these four because these are the main glycerophospholipids that can be found in almost every single cell type. The other group is, as I said, Sphingophospholipids, and one of their main examples is Sphingomyelin. Now, you might heard before of myelin sheath, which are a type of lipids that can be found surrounding the neural axons within the nervous system. Now, this is a major component of the myelin sheath, sphingomyelin. So now we will discuss the different types of glycerophospholipids. So the first type, or the first example that we mentioned, is phosphatidylinositol, PI. Now, phosphatidylinositol can be found mainly in the inner leaflet of the cell membrane. So this is the two different leaflets facing different sides. Phosphatidylinositol can be found mainly here, facing the cytoplasm. It's negatively charged, so the head of the phospholipid, this is the head, and these are the tails of the glycerophospholipids. So the head is negative. That means that these two are overall negatively charged. They can interact as a result with positively charged proteins. So proteins can attach to the membrane by interacting with the negatively charged head of the phosphatidylinositol. It can be phosphorylated in 7 different ways or 7 different combinations, resulting in the creation of something called phosphoinositides. We will discuss this within the next few slides or a couple of slides. It's important in cell signaling and lipid signaling and membrane trafficking. So it has huge importance in, as I said, cell signaling. So it can be actually metabolized and it can turn into different components, which will result in a signal within the cell itself. Maybe we will discuss this in cell signaling when we discuss the different types of cell signaling. It can move laterally and spin 360 degrees. So it can spin around itself and it can move in different directions. So 1 second it's here, another second it's in completely different part of the cell. And this applies to almost all the lipids and even proteins. So they can actually move all over the cell. They're not stuck in certain positions, except in some few examples of proteins where they cannot move for some reason. We will talk about this later. But as I said, lipids can actually move along laterally along the cell membrane. Hence the name, if you heard this term before, the fluid mosaic model. Yeah, because it's moving, the components of the cell membrane are moving all the time. So this is the generic structure of the phosphatidylinositol. We have an inositol attached to a phosphate, which is attached to glycerol, and the glycerol is attached to 2 fatty acids. Hence the name lipids. Okay, and phosphatidylinositol because we have an inositol group attached to a phosphate here. So the 7 different phosphorylated types or combination of phosphorylated phosphatidylinosytols, the first three types are referred to as monophosphates. That means that it has one phosphate attached to the inositol. So this is the inositol type that I just mentioned, which is attached to a phosphate group. So inositol can have a phosphate attached to it at three different positions or three different carbons. So it can be attached to carbon number 3, carbon number 4, and carbon number 5 individually. So if it's attached to carbon number 3, in this case it's referred to as phosphatidylinositol 3-phosphate or PI3P. If the phosphate is only attached to carbon number 4, then it's referred to as phosphatidyl4-phosphate or PI4P. If the phosphate is attached to carbon number 5, then it's referred to in this case as phosphatidylinositol 5-phosphate or PI5P. So these are monophosphates because they have only one phosphate attached to them. In case if you don't know what phosphorylation, it means the addition of phosphate. That's phosphorylation. The next three combinations or three types are the biphosphates, meaning that two phosphates are attached at the same time at two different carbons in the inositol. So if the phosphate is attached to carbon number 3 and carbon number 4, then in this case the inositol is called phosphatidylinositol 3,4-biphosphate or PI3,4P2 because we have two phosphates. If the phosphate is attached to carbon number 3 and 5, then it's phosphatidylinositol 3,5-biphosphate or PI3,5P2. The same with if a phosphate is attached to carbon number 4 and 5, then in this case we have phosphatidylinositol 4,5-biphosphate or PI4,5P2. It's also called PIP2. These are called biphosphates because, again, two phosphates are attached at the same time to the inositol. Now the last combination or type is the triphosphate. Now triphosphate means that three phosphates are attached to the inositol. So phosphate attached here, here, and here at the same time. In this case, it's referred to as phosphatidylinositol 3,4,5-triphosphate or PI3,4,5P3 or PIP3 as short form. So these are the different phosphorylated types of phosphatidylinositol. The glycerophospholipid. So the second glycerophospholipid is the phosphatidylcholine, PC. Now the phosphatidylcholine can be found mainly in the outer leaflet of the cell membrane, unlike the phosphatidylinositol, which is here in the inner leaflet. It's the most common phospholipid. So if we extract again the lipids that make the cell membrane, then 60%, as I said, of these lipids will be phospholipids, and the majority of these phospholipids will be phosphatidylcholine. Again, it can move laterally or spin 360 degrees like the other lipids. So 1 second it's here, another second it's on the opposite side of the cell, for example. It moves really, really fast. It's like fluid, hence the name fluid mosaic model. Everything is moving around. This is the generic structure of phosphatidylcholine. So it has like the same backbone as the phosphatidylinositol. So we have glycerol, phosphate, and fatty acids, hence the name Glycerophospholipids. But instead of the inositol, we have choline, hence the name Phosphatidylcholine. Now the next glycerophospholipid is the Phosphatidylserine, or PS. Now found mainly in the inner leaflet, so it's similar in terms of positioning to Phosphatidylinositol. It's also negatively charged, so the head of the Phosphatidylserine is the overall charge is negative, and therefore it can interact with positively charged proteins. That's how some proteins attach to the membrane, through this interaction, negative-positive interaction or attraction. It also, as I said, can move laterally and spin 360 degrees. So it's not locked in one position. This is the generic structure of the Phosphatidylserine. So again, same backbone, glycerol, phosphate, fatty acids, hence the name Glycerophospholipids. But we have serine, the amino acid serine, instead of the inositol and the choline, hence the name Phosphatidylserine. The last glycerophospholipid I'll be talking about is Phosphatidylethanolamine, or PE. Now found mainly in the inner leaflet, just like the inositol, Phosphatidylinositol, and Phosphatidylserine. It's the second most common phospholipid type. So after Phosphatidylcholine, we have Phosphatidylethanolamine, or PE. It can also move laterally and spin 360 degrees, just like the other lipid types. It has the same backbone, so glycerol, phosphate, fatty acids, hence the name Glycerophospholipids, or it has an ethanolamine as part of the polar head group instead of the inositol, choline, and serine. So these are the major glycerophospholipids. Now, the other groups, which is the Sphingophospholipids, one of the major examples of Sphingophospholipids is Sphingomyelin. Sphingomyelin can be found mainly in the outer leaflet, so here, just like the Phosphatidylcholine. It's quite common. It can form something called rafts with cholesterol. Rafts are certain areas or positions or domains within the cell membrane which are rich in cholesterol, proteins, and sphingolipids like the sphingomyelin. Now these have important function in terms of cell signaling, and they're called rafts because they move around the cell. They can move, these areas can move from one side to the other side of the cell. Now, sphingomyelin can be found mainly in nerves, as I said before, as part of the myelin sheath. So it has important in terms of neurotransmission. It can also move laterally again and spin 360 degrees. This is the generic structure of sphingomyelin. So we have, and that's what makes it different from glycerophospholipids, is that we have sphingosine instead of glycerol. So we can't have both glycerol and sphingosine. We either have glycerol, so in this case it's called glycerophospholipids, or we have this sphingosine. In this case it's called sphingophospholipids. So yeah, sphingophospholipids. And as part of the polar head, we have either choline or ethanolamine as attached to the phosphate group. This what makes the sphingomyelin. So it's similar to phosphatidylcholine or phosphatidylethanolamine, except instead of a glycerol, we have sphingosine. Now these are the different types of phospholipids. Now let's move on to glycolipids, which are less common lipids that can be found as part of the cell membrane structure. They can also be classified into two major groups. Again, you probably can know the difference. You can probably distinguish the difference between these two. So glyceroglycolipids, and we have sphingoglycolipids, or another pronunciation, or another way to write it is Glycosphingolipids, but I like to write it this way because in this case you can compare them to phospholipids and you can know the difference between these two. So the glycolipids, glyco, as I said before, refers to sugars or carbs. I don't know if I said that actually, but that's why it's called glycolipids. Unlike phospholipids, we have a phosphate group. Here we have sugar, no phosphate. And it's called glycerophospholipid because we have glycerol. Okay, so glyceroglycolipids can be found mainly in the outer leaflet, so with the choline and with the sphingomyelin, or phosphatidylcholine and sphingomyelin. They are rare, and they can form rafts again, just like the sphingolipids or sphingophospholipids. They are also important for signaling, and they are mainly found in photosynthetic cells, okay, like plant cells, for example. They can move laterally, again, just like the other types of lipids, and spin 360 degrees. So if we look at the generic structure, we don't have a phosphate. That's why it's not a phospholipid. We have sugar. That's why it's a glycolipid. Okay? So we have glycerol and fatty acids, hence the name glyceroglycolipids. The other type of, or the other group of glycolipids is the sphingoglycolipids, or again, you could call it glycosphingolipids. Now, they can be found mainly, again, in the outer leaflet, just like the phosphatidylcholine, or the sphingomyelin, or the glyceroglycolipids. So all of these can be found here in the outer leaflet. They are also rare, and they can form rafts with cholesterol, important for signaling, and they are similar in terms of that they are common in the nervous system, just like similar to sphingomyelin. They can also move laterally and spin 360 degrees, again, just like the majority of lipids. So if we look at the generic structure, instead of glycerol, we have sphingosine, hence the name Sphingoglycolipids. Because as I said, as a rule, you either have glycerol or sphingosine, and this determines if it's a sphingo or glyceroglycolipids or phospholipids. Now we will move on to the last classification or class of lipids that can be found in the cell membrane. Now, one of the main or major examples of sterols is cholesterol. Now, cholesterol can be found in both leaflets of the membrane. So if you look here, we have it here in the outer leaflet, or in the inner leaflet. It's quite common, so you can find it's almost in every single cell membrane, and again can form rafts with sphingolipids, and it can form rafts as well, with sphingolipids. So both sphingophospholipids or sphingoglycolipids. So there, it's important for signaling. That's what I'm trying to say. It has also importance in stabilizing the cell membrane. So it interacts with the neighboring lipids and proteins, and specifically the hydrophobic regions here, and this is the hydrophilic regions, the regions loving the water, because the majority of the inside and the outside of the cell is water. So it's important in stabilizing the membrane by interacting specifically with the hydrophobic region of lipids and proteins, and therefore it prevents freezing at low temperatures, because at low temperatures, if it wasn't for the cholesterol, this would all freeze and the membrane becomes really rigid, and therefore it will be really hard to actually to function, because ions, for example, and water won't be able to get in or out, and ions won't be able to get in and out of the cell, and even the proteins won't be able to function normally because they will be deactivated. So this is prevented by these cholesterol. So with the interactions, it keeps the fluidity of the membrane to a certain degree, and therefore it keeps it from freezing. At higher temperature, on the other hand, it keeps the membrane from boiling. So at higher temperature, all of these different components will be moving all over the place because it's really hot. But because of these cholesterol, they interact again with the hydrophobic region of the different lipids and proteins as well, and therefore the cholesterol keeps these different structures in their place, so they keep them calm to a certain degree. It also prevents excessive fluidity, specifically at higher temperature. As I said, at higher temperature, these will be moving a lot and therefore probably leaving a lot of gaps. But because we have cholesterol, then this is prevented. Therefore, it prevents increased permeability. So not everything will be able to get in the cell because of the high temperature, for example. It's again important for signaling. As I stated before, it can form rafts with sphingolipids, but it's also important in the production of vitamin D and steroids, because the cholesterol is used as a precursor. So, we can synthesize, the body can synthesize vitamin D and steroids like testosterone, for example, using cholesterol. Again, it can move laterally and spin 360 degrees like the other different types of lipids. This is the generic structure of cholesterol. So we have a hydroxyl group, which is the hydrophilic head, just like the different heads of phospholipids, where we have phosphates and inositol, for example, and the heads of glycolipids, where we have the sugars, for example. We have also the hydrophobic region, this whole region. We have the steroid rings, 4 rings, and we have the hydrocarbon side chain. So these are hydrophobic. These are the ones that interact with the hydrophobic region of the different lipids. And this one interacts with the hydrophilic region of the different lipids. So lastly, these are notes worth mentioning about the structures and functions of lipids. So the fatty acid tails of phospholipids and glycolipids can be either saturated or unsaturated. So if it's saturated, then that means it has only single bonds, like this structure here. And it's straight, as you can see. If it has a double bond, then it's referred to as unsaturated. If it has one double bond, then it's monounsaturated, and it's a little bit curved, as you can see. If it has multiple double bonds, then it's referred to as polyunsaturated, and it's really curved, as you can see. Now, these are important in terms of determining the fluidity of the cell membrane. So the more double bonds, the more unsaturated the tails or fatty acid tails of the phospholipids and glycolipids, the more fluid the membrane is. The same with the length of the tails. So the shorter the tails, the less interactions between the different tails of the different types of phospholipids and glycolipids, and therefore the more fluid the cell membrane is. So short, more fluid, more double bonds, more fluid. Some of the structures of the cell membrane are also present in membrane organelles, but in different concentrations. For example, the mitochondria has the mitochondrial membrane has phospholipids, has glycolipids, but in different concentrations. The same with the endoplasmic reticulum membrane. It has phospholipids and glycolipids, but it has very low cholesterol concentration because cholesterol, as I said, it interacts a lot with the proteins and lipids surrounding it, and therefore it interrupts protein synthesis. So it's better to have low concentrations of cholesterol, but it has also other types of phospholipids like choline, for example, and serine. So these structures that we talked about are not only common in the cell membrane or structures of the cell membrane, but they are quite common in structures of other organelles as well. So this concludes our video for today. If you like the video and the content of this video and this channel, then please like, share, and subscribe. You may also visit our website for further updates in the future. Thank you very much, and see you in future videos.